Ladies and gentlemen, good evening. Tonight I'm going to talk about something which is very close to my heart because I love wine. I've been fascinated by wine for many, many years. I did an MSc and a PhD in brewing microbiology, and I did my PhD in Belgium. And one night my supervisor invited me to his house and served a 1961 Lafite wine, which was just an exquisite wine. I've never had anything like this in my life. And right there, I switched from doing research on beer to doing research on wine. And I've done that for the rest of my life up to now. And it's just been a fascinating experience. It was more than 100 years ago that Louis Pasteur said, wine is the flower in the buttonhole of civilization. And I truly believe that. I've had some spectacular wines. You know, we know a lot of people that are wine lovers. Many of them are very well educated in wines. And we often have dinners and we sit around and enjoy a glass of wine that's 20 or 30 years old. And for 3 hours we sit and discuss this little glass of wine. It's the only glass of wine. The wine keeps on changing in the glass. It's just fantastic. And I'm just going to give you a short overview of how wine is produced. First of all, red wine, the grapes are crushed, destemmed, and then there's a fermentation and the pressing. And the white wines, the same thing, the grapes are destemmed and crushed, and then there's a fermentation. And then in some white wines, there's a malolactic fermentation. It's a conversion of malic acid to lactic acid. And most red wines undergo this fermentation. And this is done by bacteria, which are inoculated into the wine after the alcoholic fermentation. And you've got to keep in mind, after the alcoholic fermentation, there's a lot of alcohol. The nutrients in the wines have been depleted, and it's very difficult for these bacteria to start to grow. And the malolactic fermentation is used for most of the red wines and some white wines. It leads to deacidification of the wine, bacteriological stability. If you don't get this fermentation going, these bacteria can grow and bottle, and they produce carbon dioxide. When they decarboxylate the malic acid to lactic acid, then the corks come flying out and they cause turbidity. And some people believe it also leads to the development of flavor and aroma compounds in the wine. And this fermentation is conducted by Unococcus uni, the bacterium. And unfortunately, this is a very unreliable fermentation. It leads to stuck malolactic fermentation. This is the conversion of malic acid to lactic, is called the malolactic fermentation. It leads to capacity problems in the cellars. You can think if a winery sits with 100,000 liters of wine in a big tank, and this malolactic fermentation will proceed, and there are more grapes coming in. It causes major problems in wine. It leads to spoilage of the wines because the wines cannot be sulfited, so any spoilage organisms can grow, and the production of biogenic amines. And what are these biogenic amines? These are some of the biogenic amines that have been isolated from wines. And you will probably recognize things like histamine and tyramine. And a very large population, about 30% of the world population, is sensitive to these bioamines, and they get severe headaches when they drink wine. So about 30% of the wine population, or the population in the world, don't drink red wines, or mainly Chardonnay wines. And I'm unfortunately one of those people that love wine, but get severe headaches if I drink these wines. So the only way I can get by it at the moment is to drink old red wines, well-aged red wines, because these bioamines precipitate in these wines and they don't affect me that much. And I decided, you know, I love wine so much and I'd like to help the 30% of the people in the world. So I undertook to engineer the malolactic wine yeast MLO1. And this is just a picture, electron microscope picture of Saccharomyces cerevisiae, the yeast cell that ferments sugars to alcohol and carbon dioxide and flavor compounds. And this is the yeast cell, and these are little bud formations. When this yeast cell multiplies, it forms a bud on the edge, and the bud just drops off and forms a new yeast cell. And a single mother cell like this can produce about 40 daughter cells. And that's the way the yeast cells grow and multiply. A lot of people think of cloning as something mystique. The only thing cloning is really the isolation of a gene. That means you isolate a gene and you have the gene in your hand, or rather in a test tube. So I had to construct a malolactic cassette because there are two genes required. One gene is the May1 gene, which we cloned and characterized in my laboratory. And this gene is responsible for bringing the malic acid across the membrane into the yeast cell. And the second one is the malolactic gene, which decarboxylate malic acid into lactic acid and therefore do the malolactic fermentation. And we integrated this linear cassette into the genome of wine yeast. We had to develop tests to see which strains, which of the yeast cells contain this strain. And these are 4 controls where we just added lactic acid, and you can see there is one strain that we found. Eventually we found about 3 of these clones in about 3,000 colonies that we screened. It was quite a job. So we did some vinification trials, winemaking. We studied malate degradation, did organoleptic analysis of the wine produced with the malolactic yeast, and we used 2 types of juice. Cabernet Sauvignon juice with 6.21 grams of malic acid, and in the cold regions in the world, such as British Columbia, for instance, the malic acid concentration get very high. It can go up to 9 grams per liter of malic acid, which is very, very difficult for malolactic bacteria to handle. It's almost impossible. So it causes problems. It's a major problem in cool climate regions. And we also used Chardonnay from Kelowna, which had 9.22 grams malic acid in the grape must. Well, we fermented these wines, and here you can see everything was done in duplicate. This is the, let's see, the control strains with Unococcus uni, the bacterium in there. And right here you can see is a strain that we had functionally improved to do the malolactic fermentation. So within 4 days, the malolactic fermentation was completed. And the nice thing about this, the yeast is not sensitive to bioamide or to sulfites. So you can actually add the sulfites during the fermentation. It prevents the growth of any spoilage organisms and organisms that can produce these noxious compounds. Look at how long it takes before the malolactic fermentation was complete in this case, and this is where the bacteria was added. In this case, it was never completed, almost 220 days. And remember, you cannot add sulfite during this time, so any spoilage organism can grow and produce these bioamines. The wines that we produced were analyzed by sensory analysis by an independent panel. And you can see the yellow, just follow the yellow, is the malolactic yeast. It was independently done and unmarked, so people didn't know what they were tasting. So the malolactic yeast that we engineered produced the highest quality of yeast, the best color in the wine. There was no loss in the color of the wine. It had the highest fruity taste. It had the most body. You know, sometimes wines are thin, and other times when you put it in your mouth, you can feel there's some body to it. It had the sweetness and had the lowest acidity. So it worked very well. We then had to apply for regulatory approval. This was another PhD that I had to do. The FDA, the US FDA gave us GRAS status in 2003. It means that the wine is safe, the yeast was safe to use. It was about a 400-page document that I submitted. And I thought this was bad, but this was about 800 pages that I had to submit to Health Canada and Environment Canada. It took quite a long time. We've applied to the South African regulatory body, and it was approved by the regulatory body, but the wineries asked that the wine not be used because they were concerned because they export so many wines to Europe and it hasn't been approved there. And a lot of the people there have been scared by genetic engineering. And we're in the process of applying to the Australian regulatory body and also to the European regulatory bodies. To summarize, we have a fully functional malolactic wine yeast. We've sequenced the DNA introduced, no antibiotic marker genes in the yeast. It is substantially equivalent to the original strain. It is genetically stable. A residual population of malolactic yeast is not a concern to wineries. Many wineries asked, well, what happens if we use it once? Will we always have malolactic fermentation? The answer is no. You can just rinse a tank and you will not have any problems. There's a clear health benefit to consumers, 33% of the world population. and definite advantages to wineries. And the benefits to winemakers, it's an efficient malolactic fermentation. They won't have any problems. No stuck malolactic fermentations. It'll prevent the growth of spoilage organisms. It enhances the red color in wine, solve capacity problems, prevent the production of bioamines, and it's a very cost-effective process. We licensed this technology to a French yeast company of all people. You know, the French, the Europeans are so anti-GMO, and yet they said there's a huge business there. It's been approved in Moldavia since 2004, and the US since 2002 Moldavia, 2004 in the United States, and it's been used in Canada since 2006. And it's taken me 15 years to do this work. We did a lot of work. We did 7 years of the construction of the yeast and then 8 years of testing. These are all of the PhD students that have worked on this yeast strain. And that's what I have to tell you. This is very superficial. You know, I worked for 7, 8 years. We have a lot of data, but I didn't want to present all the hardcore scientific data here because I know a number of you are not scientists and it will be hard to explain it in such a short period of time. But if you have any questions, feel free to ask.